Hello everyone, this is Fahim Ahmed from Edexcel Exam Essentials. We will be solving this paper from Biology Unit 4, and this paper is from the year October 24. This video is going to be for people who are struggling with how to get on started with the question papers, and we are going to cover all of the process in details. So without wasting any more time, let's just get started. First question. He said about that very little of the light energy falling on a field of plant is being absorbed by the plant and transferred to the next trophic level, right? So suggest two reasons behind this. Suggest two reasons why very little of the light energy is absorbed by the plants. Okay, right. So in the chloroplast of the plants, we have these pigments known as chlorophyll, right? So these pigments. So there are these different type of these pigments. For instance, chlorophyll A, then chlorophyll B, and so on, carotenoids and so on, right? So what they do is they are responsible for absorbing different wavelengths of light from the sun and then convert it into chemical energy, right? So for the time being, understand that because it's a way more complicated process. And for this question, we do not need to know what they do with this light energy. Like the electron gets excited and so on, right? But for the time being, just understand that there are these different sorts of these green pigments, there are these different sorts of these photosynthetic pigments that is present in the chloroplast of the plant, and they're responsible for absorbing different wavelengths of light from different wavelengths of light. And yeah, I mean, this is what you need to know about all of this is what you need to know for this question. And another thing would be there are these other pigments as well, and there are these other pigments such as carotenoids and so on, and each of these pigments absorbs light of a specific wavelength. Most of them would absorb light in the blue and the red part of the spectrum. And most of the plants would not absorb the green light as well. They would try to reflect off the green light, and hence the plants appear to be green. So yeah. So what could be some of the reasons that not all of the light energy is absorbed by the plants? So there could be a couple of reasons for this. So first thing is that all of these pigments, right? Like there is this different wavelengths of light that falls on these pigments, these photosynthetic pigments. So not all of these wavelengths of light is being absorbed by the plant, could be one reason. And apart from that, the green light, as I said, the green light is directly reflected off from the leaves of the plant as it cannot get absorbed by the photosynthetic pigments of the plants. And another reason could be that the light energy or the light that is falling on the plant, like suppose this is the plant, right? So the light that is falling on the plant is not falling on the leaves. Like on the chlorophylls, photosynthesis takes place at highest rate, right? And produces a glucose, and then they can use it to carry out their metabolic activities and so on, and create a new biomass, and that new biomass would be transferred to the next trophic level, right? But when light falls on the parts of the plant that do not carry out photosynthesis, then no glucose would be generated, and hence no energy would be generated to create a new biomass that can be passed to the next trophic level. So yeah, this is one of the other explanation for this question, and this is what I have written over here. Now, in the next question, it says something about it's basically a basic math, right? So it says that in American study, light energy falling on 1 square meter of a field is in 1 year was recorded to be this much of kilocalories, right? And 1/15 of this light energy was converted into plant biomass. 1/15. So 1/15 of this thing was converted into plant biomass. Calculate the number of calories of light energy converted into plant biomass. So it's basically a simple math. You do 1/15 of that entire number to get the biomass that was converted into plant biomass. Light energy converted into plant biomass. So and it said that only 6 kilocalories become animal biomass. Calculate the number of kilocalories of energy that become animal biomass. Again, and 1 kilo. So it basically is an unitary method where 1 kilocalorie represents 4.2 kilojoules, and so 6 kilocalories would represent 4.2 times 6 kilojoules. So yeah. And it said that in another field, this much of kilocalories of energy was converted into plant biomass. Okay, only 25 kilocalories of this energy was passed to the highest trophic level on the in a food chain, right? Calculate the percentage of energy that was passed on to the highest trophic level. So yeah, so I mean it's again a basic maths. So you have the 25 that was passed to the next trophic level by total amount of energy kilocalories available times 100, so you get percentage of that. Right, so moving on to the next question. Yeah, and you can find the details of this question from I believe the second chapter of your book about chlorophylls and everything and photosystems. But for this question, you didn't need to have an idea of that photosystems at this point. So yeah. He said that broccoli is a vegetable plant that is grown as a crop in many parts of the world, and broccoli is susceptible to a disease called clubroot. Okay, genetic engineering can be used to produce clubroot resistant varieties of broccolis. Okay, and genetic engineering involves inserting resistant genes into the cells of the broccoli plants. Okay, the resistant genes were obtained from linseed plants. Okay. Scientists can use DNA profiling to identify broccoli plants that have been successfully genetically engineered. Okay, right. So, I mean, this question is about producing recombinant DNA and everything. And yeah, it said here that the broccoli plant is susceptible to a disease known as clubroot, and if we can insert genes from the plant of linseed that is resistant to this clubroot disease, so yeah, the broccolis would be resistant as well. Now this question asks us to find the broccolis that have been genetically modified. So yeah, acha. Right. So it said that some samples of DNA were extracted from plants subjected to polymerase chain reaction before analysis, right? So why do we basically use a PCR, like polymerase chain reaction? So polymerase chain reaction. Give me a moment. Microbiology. Sorry. Polymerase chain reaction, right? So this is the overall procedure of this polymerase chain reaction, and we do this to amplify our DNA sample. We amplify our DNA sample via this procedure. So what we try to do is we try to mix the sample that we want to clone with the sample of primers, with the samples of primers and four bases in sufficient amount and a buffer solution and DNA polymerase enzyme, right? And then we put all of this in a PCR machine and heat it to a temperature of 90 to 95 degrees Celsius. This causes the strands of the DNA to separate. And once this strand of the DNA gets separated, we then decrease up the temperature. We then decrease up the temperature to uh, we then decrease up the temperature to around 50 to 60 degrees Celsius, and this is when primers join to the strands of the separated strands of the DNA, and then we can increase the temperature to 75 degrees Celsius so that DNA polymers can get activated and bind with the primer and carry out the process of synthesis of producing complementary DNA in the respective directions, right? So primers are nothing but basically a short sequence of DNA that enables DNA polymerase to begin the process of synthesizing complementary DNA. I mean, in short, it basically tells why to start the synthesis of the DNA, complementary DNA from right. So explain why PCR was used on this sample. First thing was that it was used to amplify the sample, increase in number of the sample, so that the samples could then be identified using techniques such as gel electrophoresis to identify that which plants were genetically modified and which were not. So I have basically written this down in my answer over here, so you can just take a look at it. Right. So tagged DNA polymerase is used in PCR. Describe the role of tagged DNA. So first thing is that it binds to the primers, as I said over there, and then it starts the synthesis of complementary strands of DNA. I mean, it basically tells like where to start the synthesis of the complementary strands of DNA from, so that the strands of the DNA could be replicated again and again. I mean, the main aim of PCR is to produce a higher percentage of the sample of the DNA, so that they could be used in a lot of techniques such as forensics and so on. Right? And after PCR, as I said, after PCR, gel electrophoresis was used to separate DNA fragments. Which type of enzyme is used to produce the DNA fragment? So this is going to be restriction in the nucleus. This enzyme is going to be restriction in the nucleuses. The next question is about the results of gel electrophoresis were used to identify plants that contain resistant genes, right? Deduce the type of plants that have supplied the DNA samples for the analysis. Okay, so it said about the type of plants that we can use for the samples for the gel electrophoresis, right? So before we get started with this, you need to know something like how do we identify that a plant has been genetically modified or not, right? So to do this, we place the samples of the DNA in a wheel. Suppose this sample is from a genetically modified plant, GM plant, genetically modified plant, right? And this is produced from these bands produced by using this gel electrophoresis technique. I mean, it's a detailed process, but you do not need to know much of it in this question. So I'll not be going through all of this, but I'll be giving a brief overview of this. So suppose it's the DNA sample from a genetically modified plant and the DNA strands get separated. And here there is this DNA sample extracted from other genetically modified plants as well. Other genetically modified plants also. So where the bands would overlap with each other. So if the bands overlap with each other, suppose these bands are overlapping with each other, right? So this would suggest that they comprises of the gene, they comprises of the same gene, same resistant gene and so on. So the question there is about this thing. It said that identify the DNA samples that we could have used for this technique, right? So for this, I could have used the DNA from the genetically modified plants so that I could identify the other genetically modified plants by comparing their DNA strand or the height of the DNA strands, either they're same or not. I could have also used the original broccoli plants and compare its band with the genetically modified plants to identify the genetically modified plants as well. And then I could have used the linocytes. The linocyte remember that the linocytes they produce the genes that were resistant to club groups, right? So I could have also used the linocytes to identify the resistant genes by seeing their bands again. So yeah, I have basically written this down over here. Moving on to the next question, it said that a person's immune response to viral infections involves T killer cells, right? Cells infected with viruses present cells infected with viruses present antigens, the T killer cell, right? Um, Peptide fragments bind to MHC complex to form antigen MHC complex. This takes place in the rough endoplasmic reticulum. The complex is then transported to the surface of the infected cell. Describe how antigen MHC complex is transported to the surface of the cell from rough endoplasmic reticulum. Right. So this question is basically from AS biology. How protein is transported, right? So suppose you have this rough endoplasmic reticulum over here. So rough endoplasmic reticulum. So the proteins, after they are synthesized, gets packed into these vesicles, from the rough endoplasmic reticulum and moves to the Golgi apparatus. So suppose this is the Golgi apparatus. I mean, this is a rough drawing, excuse me for that. Suppose this is the Golgi apparatus, right? And the vesicles then fuses with the Golgi apparatus and releases its content inside the Golgi apparatus. And once inside, the contents like the proteins, the antigen MHC complex in this context gets modified. And after that, from this protein, this antigen MHC complex get packed up into secretory vesicles, into the secretory vesicles from this Golgi apparatus, right? And from there, it then fuses with the cell surface membrane of a cell and releases of its antigen MHC complex on the cell surface membrane of the cell. Right. So, I mean, this is the process that I have described over here. So, yeah, we'll take a look at it. The next question is about an electron microscope with a resolution of 3.7A can be used to study antigen MHC complex, right? So 1 equals to 1 into 10 to the power 4A. Calculate the resolution of this electron microscope in nanometers. Calculate this in Acha. All right, so just remember this. I'm going to show you a technique that can be used in either physics, chemistry, or biology, right? So whenever you're trying to add a prefix, you divide with the prefix value. And whenever you're trying to remove a prefix, what you do is you multiply with the prefix value, right? So in this question, what I can see is calculate the resolution. First of all, I have to calculate the resolution, and in nanometers, I have to calculate this resolution, right? So 1 nanometer represents 1 into 10 to the power 4A. I have written that. So 1A would represent this, and 3.7A would represent that, right? This is in nanometer. This is in micrometer. Sorry, use micro, right? How do I convert this into nano? So I have to remove this micro and add a nano in, right? So to remove this micro, I will multiply this thing with 10 to the power minus 6. And to add a nano, I will divide this divide this thing by 10 to the power minus 9. This is how I get to this answer. Right, so state what is meant by a resolution. So what is meant by a resolution? It means that the microscope can identify between two separate lines that are 3.7A or 0.37 nanometer away. So what basically is this resolution, right? So suppose there is this dot, right? Or suppose there is this line, right? However, in microscope, this thing, I mean, here in my screen, it is appearing as a single line, but I mean, as we try to get closer and closer, the lines would start to separate. So the distance between these two gaps that a microscope can identify to able to distinguish them as two separate lines is what we refer to as resolution. Explain why antigens need to be presented to T killer cells in a person's response to viral in a person's response to viral infection, right? So explain why antigens need to be represented on T killer cell in a person's immune to viral response, right? So this thing is from immunity, right? So here, just take a look at it. Suppose this is the antigen, this is the cell representing the antigen MHC complex. Antigen MHC complex, right? The T cell then identifies this, the T cell then identifies this thing, this antigen, and binds to it. And once the T cell binds to it, the T cell gets activated and gets divided into two sorts of it. One is T helper cells and the another one is T memory cells. T memory cells is responsible for providing immunological memory, and uh, T helper cell is involved in the activation of B cells or in the activation of plasma cells to produce antibodies responsible for producing infected cell and producing antibodies and so on, right? So I have written this here. Yeah. Then explain how the action of T killer cell results in a person recovering from a viral infection, right? So this thing is from the cell mediated response, not the humoral response of the immunity. So uh, um, um, right, so take a look at this here. So the thing that trying to say is that the T cells would bind with the antigen and then create a hole on its membrane. Suppose this is the cell containing the virus, right? So the T cell would bind with this and would create a pore on the membrane of the virus particle. And once a pore is created, then water and oxygen ion can move into the cell containing the virus particles, move in and out, and this would cause the cell to rupture. And once the cell gets ruptured or the cell gets damaged, the virus particles are then released. The virus particles are then released into the surrounding tissues and cells. And once the virus particles are released, the macrophages the macrophages then goes to the side and then engulfs it. The macrophages. Why do the macrophages goes to the side that comprises of this virus particles, right? Because this virus particle produces of some sort of chemicals. You do not need to know details of it, but they produce of some sorts of chemicals that attracts these macrophages to that affect to the side that comprises of the virus. Yeah, I have written it down over here, so you can take a look at it. Moving on to the next question. Okay. This question is from forensics. All right, so it said that the time of death of a mammal can be determined by a number of different methods, right? Insects can be used to determine the time of death of a mammal. Which term is used to describe the use of insects determining the time and place of death of a mammal? So it's known as forensics entomology. In entomology, forensics entomology. Um, Which of the following factors provide information about the time of death of mammals? So, species of insect, yes. Maggots, beetles, blowflies, yes. Size of the larvae, yeah. Maggots and so on. Stages of development of insect, yes. So, it's going to be three of these. So, to give a basic idea about this process, let me just take you into it. Sorry. Let me just take you process of decay, right? So, first of all, as a person dies, the body acts up as the soil. The body acts up as the soil, and the first colonizer starts to appear after catabolic enzyme decomposes the guts and the surrounding tissues of it. I mean, this makes the condition less harsh for the pioneer species or the first colonizers to appear. And the first colonizers are anaerobic bacteria that requires less oxygen and can work in lactic acid rich environments of the muscles, and they decompose faster and makes the habitat more suitable for the upcoming species. And then we have these blowflies it comes into play, and they lay up their eggs that hatches into maggots, and this releases of digestive enzymes that try to liquefy the cells of the body. And once the maggot becomes adult, the cycle starts to begin again, right? And then beetle starts to appear and lay eggs, and the larvae feeds on the maggots itself, right? And gradually the body gets dried up, and that's when the first colonizers starts to disappear, and species that has more stronger mouth or jaw parts starts to appear to feed on the remaining connective tissues and muscles and bones, right? This is the entire process of decay. Yeah, the body temperature of a human corpse can be used to determine the time of death, right? Okay, yeah, it does. Which is the most accurate method of measuring the body temperature of a human corpse to determine the time of death, right? So this would be pushing a temperature probe into the liver, because the liver starts to keep on continuing to do its metabolic activities after a person even dies, and yeah, it releases a heat, and then it starts to it gradually starts to cool down. So liver would give the most accurate measure of the temperature instead of placing it on the forehead or any other parts of the body, as it is more exposed, as all of the other parts would be more exposed to the environment and hence would cool down faster. The time of death can be estimated by determining the fall in body temperature, which row of table shows how more clothing and higher ambient temperature would affect estimated time of death. So suppose this is the person, right? And if the person is surrounded with more clothes, it would act as an insulator and would prevent the heat from the person to get to the surrounding, right? And another thing is that ambient means the surrounding temperature, right? So if surrounding temperature is greater, then heat from the person would diffuse to the surrounding less rapidly. Less rapidly, it would diffuse less rapidly. You have to know this thing that um, energy dissipates or distributes from an area that is higher to an area that is lower, right? However, if the temperature is higher as well, the outside, then it would get to the surrounding less quickly. So yeah, I have chosen A for this. A for this. The extent of muscle contraction, rigor mortis, that can be used to estimate the time of death, right? So what basically is this rigor mortis, right? So after a person dies, so basically this is the entire process of rigor mortis. So after a person dies, they're to keep their muscle in a relaxed state, they need the muscles need to be supplied with energy that can be provided to the muscles by the breakdown of the stored glycogen to maintain the muscle in the relaxed state. And once all of those energy gets used up, the muscle then starts to contract. And once the muscle starts to contract, the body of the person enters into a state known as rigor mortis. This can last between 2.4 to 36 hours, and after that the species of organism will start to produce enzymes, or would start to break, or would start to feed on the muscle cells, and it would again get back to its relaxed state, right? This thing is related with unit 5. This thing is related with unit 5, with how the muscle contracts. So in this part of biology, you'd understand, or you'd get a detailed overview of why the muscle to be in a relaxed state needs energy. I mean, that's basically okay, right? So there are some of these changes. ATP, ATP, lactic acid, and pH, right? So these are some of the changes that takes place within muscle after the death of a mammal, right? So I can say that lactic acid increases, ATP decreases, pH decay. Okay, so ATP decreases because energy is needed to maintain the muscle in a relaxed state. Lactic acid increases because the muscle carry out anaerobic respiration, and the pH also decreases for this anaerobic respiration as well. So explain three characteristics as shown in the graph user. Yeah, so this is what I have written over here. You can take a look at it. And said that The graph shows changes in the extent of muscle contraction with time after death. Explain why the extent of muscle contraction by itself may be inaccurate, may give an inaccurate time of death. Use information. Okay, right. So muscle contraction and the time of death would vary, right? Would vary because rigor mortis last starts from 2.4 hours and lasts up to 36 hours. So after that, this entire procedure would become inefficient. It wouldn't work. And apart from that, muscle cells would have different muscle cells would have different glycogen stores, and as a result, would affect the degree of contraction. Some muscles can contract would be able to contract for a higher periods of time, and some for a lower periods of time. And apart from that, calcium ion that is needed for muscle contraction. Calcium ion that is needed for muscle contraction may run out of supply as well, and hence, yeah. So this is just give me a moment. Just let me do a recap on that. Um, so there are these two. So there are these two components, actin and filament, right? So a muscle contracts due to the interaction between the actin and filament to form actomyosin cross bridges, right? But for the time being, just understand that this is actin, right? This is this is myosin. This is myosin. These are beads of myosin. There are these two chains of beads of myosin that are connected together by a chain known as tropomyosin. And on the surface of tropomyosin, we have a molecule known as troponin. So calcium from sarcoplasmic reticulum binds with this troponin and opens up this and pulls away this layer. And once it pulls away these layers, the myosin binding sites, there are these myosin binding sites on the actin gets exposed, and myosin heads can bind with this and result in muscle contraction. So if the calcium ions are short in supply or they get used up, the degree of muscle contraction would be affected as well. Although this is U4, I am finding a lot of concepts that are being related with Unit 5. Okay, succession, right? Succession can result in formation of different types of habitat. State what is meant by the term succession. So succession is the procedure by which communities of organism colonizes an area and over time are replaced and changes over time. So, I mean, basic definition. And said that which of the following describes a habitat? A group of interactive organism. Now, this is the definition of ecosystem. A place that meets the environmental conditions an organism needs to survive. Here's the definition. We basically know that a common definition for habitat is a place of the ecosystem where an organism lives, right? But for higher end of biology, this is the definition for an habitat that you have to understand. Right, so succession takes place as glacier retreats, or it means glacier melts. As ice melts, bare rock gets exposed. The graph shows changes in numbers of three different types of plants: alders, spruce, and hemlocks with time after glacier that has been retreated. Right. So, oh, this is an asterisk question. This isn't an asterisk question. It said that explain. Let me just take a look. Oh yeah, this is an asterisk question. So explain the changes in the number of types of plant with time after glacier has been retreated. Right. So what I could see from here is that the numbers of alders and spruce increases and then decreases. However, the number of hemlock increases and then remains constant. Right. So from here, and both of them starts from 0, alders and spruce, right? However, for hemlock starts from 20. So what I could say is that Uh, Alders and alders and spruce are the first to colonize the area and breaks down the bare rock to form soil and results in soil formation. This is the first thing that I could say. Then I could say that the increase. This is for spruce, and this is for alders. So another thing is that I can see is the increase in alder is more rapid than in spruce. The increase in alder is more rapid than in the spruce. This could be because of less competition from the spruce for nutrients and everything. Uh, then I could see that the decrease in the alders is greater as well. This is because of the competition for nutrients and resources with the spruce. And there is this another thing is that the alders number drops to zero, drops to zero, which means that they could undergo extinction because of some sorts of a disease that wiped their entire population. For hemlock, this remains constant. This could remain constant because um, there isn't much competition from spruce and alder for nutrients, and also the numbers of spruce and alders decreases as well, which further reduces the competition for hemlock, and hence their number remains constant. Right, so these are the stuffs that I have written down in my answer. So yeah, let's just take a look at it. Yeah, these are the answers. The pioneer species breakdown routes. Alders and spruce are first to appear as early soil is suitable for them. Alders and spruce increases in number as the plant reproduces. Apart from that, there is no predation. Alders reproduce faster than spruce as there is little competition from other plants. From 80 years onwards, hemlock remain constant at 18. This is because their number is supported by the environment. Alders and spruce decrease in numbers due to availability of nutrients due to availability of nutrients and diseases right? And then decrease in alder is greater than in spruce due to increased competition with spruce for water and mineral ions. And alders remains at zero, and that could possibly be due to an extinction because of a disease. So I've written that. All right, everyone. So a quick announcement before you continue our class. So basically, before exam sessions, either it's October 25, June 26, May 26, or October 26, we launch two signature courses, question paper and marathon course, high impact crash course. To know more, a short video will pop up on the screen. Please have a look into it if you're interested. You'll also find full details in the description box below. And if you want to know even more, feel free to WhatsApp us or message us on our Facebook page known as the Exam Essentials. Thank you. Right, so continuing with question number 7, said that respiratory syncytial virus, RSV, usually causes mild cold-like symptoms such as mild cold-like symptoms from which most people would recover, right? Infection with RSV can be serious in young children and in older people, elderly one, okay? Currently, there are no approved vaccines against RSV, although there are some in clinical trials, okay? They're in the stages of development in short. The graph shows the number of people who had to go to hospital in one country in 1 year. The graph also shows the number of deaths in children under 5 years old and in older people in the same country that year. Okay. what proportion of children under five in hospitals who are 6 months old. So this is the proportion of the graph that represents children with ages of 6 months, right? So how do we find this, right? So to find this proportion, each of this box represents 0.1, 0.1, 2, 3, 4, 5, right? So here it is going to be 0.1.8 and the upper is going to be 3.3. So 3.3 minus 1.8 times 10 to the power 6 divided by total number of people, total number of children would give us the proportion. So I have done that and received up my answer. That's 5 by 11. Right, so which should which should be more appropriate scale to use on the y-axis to show that the ranges of values of number of dates that could be seen clearly. So whenever you have large values, so these values are large enough, right? So whenever you have large values, you use graphs of logarithmic scale. You use graphs of logarithmic scale. I mean, you should know this by this point, right? And you should have this memorized. Like whenever you use larger values, use logarithmic scale to simplify the values, right? So it said that in one trial, people received new vaccines against RSV. The results showed that the vaccines protected against two subtypes of RSV. Two subtypes of RSV. Give two reasons why one vaccine protected against two types of RSV, right? So let's just get into vaccines for a bit. Antibiotic resistance. What is developing, right? So let's just get into antibiotics for a bit. Give me a moment. Right. So how does vaccination works, right? So vaccine comprises of these attenuated or dead strains or attenuated particles, and once it is inside the host cells, the T killer cells, and once it is inside the host cell, the T killer cell can recognizes that, binds with that, and as it recognizes it and tries to destroy that pathogen, right? So yeah, this is how vaccination works. So attenuated virus particle and yeah, lymphocyte in the lymph system. Mm, yeah, that is how basically vaccination works, right? So one vaccine could have, as it as one vaccine is effective against the two subtypes, it means the two variants of the virus. It means that it could possibly mean that both of the variants would have the same antigens that the T killer cells could recognize. Or another possible reason would be there are these two attenuated virus or two inactive or two deactivated virus, two deactivated strains of those RSVP virus, so that the body produces two types of antibodies. So this could be two possible answers. Both subtypes would have similar antigen, or the vaccine could have two type of deactivated virus that leads to the production of two different types of antibodies. C1. In another trial, pregnant women were given a vaccine against RSV. The vaccine did not cross the vaccine did not cross from mother into the development of fetus. Okay, as a result of passive immunity. As a result of passive immunity, the babies of these pregnant women were born with high levels of antibodies against RSV, right? Explain how passive immunity would have developed in these babies before they were born, right? So uh, you need to link two ideas here. So first of all is about the B cells and T cells, like the activation of B cell and T cells to produce the B cells and T cells to produce these memory cells, right? So activation of B cells and T cells to produce memory cells for giving them immunological memories and everything. And another concept that you have to link over here is transfer, developing immunity, right? So natural passive immunity, right? So in the case of a newborn babies, they do not produce up their own antibodies. Their antibodies gets transferred from the mother via placenta. Also, breastfeeding will transfer this immunity, right? And yeah, This is how the antibodies gets transferred to the fetus. However, there are these two type of immunities, that is active and passive, right? In active immunity, the antibodies or memory cells are produced by the immune cells of the body itself, whereas in passive immunity, the body do not directly produce the antibodies or memory cells against the pathogen. They directly or indirectly depends on another source. So this is linked with this question. Say that how this, okay, right? So the vaccine provided with the mother which is source of those attenuated virus particles, and this triggered immune response and lead to the formation of B and T, activation of T lymphocyte and production of B memory cells and everything. And then this antibodies pass by a placenta to the fetus, and then this remained in the fetus until they were born. So this is what I have written on it, right? So this is what I have written on this. Right, moving on to the next question. The scientists running this trial claim that the vaccine was 85% success at protecting babies against RSVP infection that required medical attention. Okay. Successful 80% at protecting babies that required medical attention. Okay. Describe how the data could have been collected to support this claim. Okay, so it's a question about data collection. So how we're going to collect this data is you vaccinate the women that were pregnant, you monitor the babies of the vaccinated pregnant of the vaccinated women over a period of time, and then you record the number of babies that needed with the vaccine that needed medical attention, and the number of babies that does not needed the medical attention, and you can find over it like that needed medical attention divided by total times 100. This is what I've written over here. Data collection. Right, it said that leaf scald is a disease of sugarcane caused by bacteria. Okay, the disease can be found in the xylem vessels. In the disease, the bacteria found in the xylem vessel, they're involved in water transportation and release chemicals, one of which is called albicidin. Some of these chemicals stimulate the xylem vessel to produce sticky gum. Sticky gum. Albicidin inhibits chloroplast development as well. So suppose this is the xylem vessel, and it comprises of this bacteria, and this produces a chemical known as albicidin. And albicidin causes sticky gum to develop on the xylem vessel. So this could impact the water flow, overall water flow, right? This could impact the water flow of the plant. So plants would not get water. And if it doesn't uptake water from the soil, it would be deprived from minerals, ions as well, right? All of the concepts of IGCSE. So basically in your A2, you have to combine all of the concepts that you have learned in AS, IGCSE, and A2. All of the three concepts are combined together. It's a mixture of all of the three concepts from all of these classes. Yeah. So explain how infection with this bacteria results in death of leaf cells and eventually the whole plant. So first thing is that chloroplast. It inhibits the development of chloroplast, right? And hence photosynthesis cannot take place. Also, as it forms sticky gums in the xylem vessel, it would hamper the uptake of water. And we know that mineral ions are dissolved in water as well, right? So mineral ions such as nitrates and so on would not get to the parts of the plant that needs them to make up amino acid and hence protein for the plant cells to make up new plant cells, right? And hence new cells cannot be formed, and as a result they would die as well. And apart from that, as there is no net production of glucose, it means that they would not be having enough ATP for carrying out their metabolic activities. This is what I've written down here. Let's just move on. Albicidin is an inhibitor to DNA gyrase or something, right? So this enzyme can be found in bacteria. So the main function of DNA gyrase is that bacteria do not have a proper nucleus, right? That like humans have their DNA in this nucleus, like the chromosomes comprises of the DNA thing, right? But in bacteria, the DNA can be found in the cytoplasm in a circular type of route, right? So DNA gyrase is responsible for the supercoiling of this bacterial DNA. Right, it binds to DNA and causes the DNA to supercoil. DNA to supercoil and process information needed to cell to function properly, right? Describe two differences between the structure of DNA that can be found in prokaryotic cell and can be found in humans, right? So in prokaryotic cell, as I said, the DNA are supercoiled and everything, right? But in human, the DNAs are linear because they can be found on two chromosomes. The DNAs are linear. And another difference would be DNA in prokaryotes comprises of more phosphodiester bond than DNA in human cell because in prokaryotes the DNA are supercoiled and everything, unlike humans. And also there is this, remember those 3 prime and 5 prime, 3 prime and 5 prime concept. So the 3 prime and 5 prime concept. So uh, prokaryotes do not have 3 prime and 5 prime, whereas humans do. I mean, if you did forget about this 3 prime and 5 prime concept, then make sure to comment below because I have videos linked to that. Albicidin is being developed as an antibiotic to human pathogens, right? So that's why albicidin can be used as an antibiotic. So albicidin can be used as an antibiotic, right? So it affects the supercoiling of the bacterial DNA. As a result, the DNA becomes inactive and cannot result in division or affects their ability to growth, right? So their DNA, in one word, becomes ineffective and hence do not support their functioning of the overall functioning of the cell. And apart from that, our human cells do not have the enzyme DNA gyrase, so this albicidin would have no impact on us as well, and hence it is going to only affect the bacterial cells. Explain the importance of developing new antibiotics such as albicidin. Okay, so first thing is that the viruses and the pathogens or the bacteria are becoming increasingly resistant to our existing antibiotics, and if no new antibiotics are developed, then we could not save our patients from these deadly pathogens. This is what I've written. Moving on to the next question. Phytoplankton. Okay. Phytoplankton are single-celled eukaryotic organisms that can photosynthesize and they live in the oceans. Okay. Coccolithophore is a, I will call this a C. C is a phytoplankton that enclosed by players in calcium carbonate. Okay. The photograph shows one species of C. This is its biological name, E. Huxley. Okay. This of calcium carbonate. This of calcium carbonate. In one drop of seawater, there are between 1,000 to 10,000 E. Huxley. The diagram shows the dimension of it, right? So this is the diameter of the cell thickness. So to find the radius, so this is the diameter, right, and the thickness. So that time, so to find the radius, suppose this is the diameter, right, and the half point of the diameter is going to be the radius. Suppose this is the radius, from here to here is going to be the radius. For radius, we have to include the thickness as well. So we have to include the thickness as well because radius is from the center to the perimeter or to the circumference. So from the center to the circumference, not up to here, from center to here. No, that wouldn't be the answer. So yeah, and then say that calculate the volume of this U. So yeah, convert that. Uh, dividing 50 by 2 would give us the 50 divided by 2 would give us the diameter, and then you add 12 to it, and the answer should be in millimeter cube, right? And here it is in micrometer, right? So 37 micrometer needs to be converted into millimeter, right? So 37 into 10 to the power minus 6 divided by the prefix of milli হচ্ছে 10 to the power minus 3, right? This is the prefix of milli. And as I said that if you want to remove something, you have to divide by the value, you have to multiply by the value of the prefix. And if you want to add something, you have to divide by the value of the prefix. And this will give us this answer. Um, Scientists think that Huxley may have an impact on global warming and climate change. Okay. Huxley are continuously producing plates of calcium carbonate using carbon dioxide from the seawater. Dioxide from the seawater. Okay. Huxley one of these Huxley can contain 12 plates. It takes 75 minutes to produce one plate. Calculate the number of hours taken, right? So to make uh, one plate, it takes up 75 minutes, right? But you have to convert this into hours, and to do that, you have to divide it by 60. And so therefore 12 plus how many hours does it take in basic unitary method? Yeah, 15. And then it said that some scientists produce some of the plates produced by Huxley are shed into ocean waters. Are shed into. The scientists have discovered that the plates shed into ocean waters can attach to other Huxley. Right? Describe how the scientists could show that the plates could attach to other Huxley. So what we could do is we could put a fluorescence tag on these shed plates. Right? We could put some of these fluorescent tags on this calcium paste so that they can grow under microscope, right? Or under UV light, right? Right. And then we incubate this shade plates with um, E. Huxley for a period of time, and then under microscope we observe the plates that grow to identify that the either they attach to other E. Huxley cells or not, the shade plates. Yeah. I mean, you can also use gene markers, to be honest. So I'm going with fluorescent dye, keeping it simple. And the last bit of it, right? So he said that the plates can carry viruses that infect E. Huxley. The infected E. Huxley produce a glue that sticks many of the cells together and form a marine snow. The marine snow is very dense and sinks to the bottom of the oceans, right? Dense and sinks to the bottom of the oceans. Okay. Explain the possible impact that Huxley could have on global warming and the effects on climate change, right? So we know that it absorbs up the healthy Huxley absorbs the carbon dioxide from the from the ocean water, right? So it would cause atmospheric carbon dioxide concentration to decrease and hence reduce the effects of global warming, such as polar melting of polar ice caps, and then rising sea levels, flooding, then seasonal change and everything, right? However, apart from that, they can also form clusters. They can also form dense clusters, and as they go underwater, right? So as it form dense cluster, it would prevent the entering of sunlight. It would form a layer that would prevent the entering of sunlight to organisms that requires the sunlight, such as seaweed, to carry out this photosynthesis. Apart from that, as this euxide get decomposed by bacteria or decomposers, it would release up this carbon dioxide that will contribute to overall carbon dioxide of the atmosphere as well. And yeah, this is this, and I have written this down here. All right, so moving on to the next question and the last question of the paper. Light is essential for photosynthesis, right? Explain the role of light in light dependent reactions. So The light-dependent reaction of photosynthesis takes place on the thylakoid membrane. On the thylakoid membrane. This is where light-dependent reaction of photosynthesis takes place, right? So remember in the beginning of the question, I said about photosystems and everything and so on, right? So suppose this is this photosystem, this is the electron transport chain, this is another photosystem, right? So light falling on this photosystem causes these electrons to jump to higher energy state, and then it moves to the electron transport chain. And this is in the stroma. And this is inside thylakoid. And this is inside thylakoid and this is inside stroma, right? So in stroma, there is a higher concentration of H plus ions. And suppose there is this ion channel known as ATP synthase. There is this ion channel. There is this ion channel. So what actually happens in the light dependent reaction, right? So the electron gets excited due to light hitting it and gets transported into this electron transport chain. And as a result, the electron transport chain have enough of this energy to pump H plus ion in the inside of the thylakoid. As a result, concentration of H plus ion inside the thylakoid increases, so they passes to the stroma via diffusion through the ATP synthase ion channel to the stroma again. And once they try to do this, and once they do this, once they do this, ADP gets converted into ATP. Then the electron then moves to the next photosystem and hence gets excited, right? And once it gets excited, there is no other photosystem. So a coenzyme, NADP, NADP takes up this electron. Where is that? Takes up this electron and a hydrogen to form reduced NADPH, reduced NADP. So one of the electron is lost, right? To replace this electron, plant would need sunlight to break down water, to carry out the photolysis of water. This is one of the need for the importance of sunlight. It results in photolysis, breakdown of it. Light is needed to excite electron that moves it and causes the production of ATP and everything, right? So this is the entire process of light dependent stage of photosynthesis, and I have written it down over here. Oh, in the entire process of light dependent stage of photosynthesis, and why do we need light for this dependent reaction, right? So this represents the entire process of the light dependent reaction, and the ATP that I said produce is needed in the light independent reaction that takes place in the stroma. The photograph shows small flowering plants found in Europe, Asia, and Africa. The effect of wavelength of light on cray's was investigated. 3 groups of crays were treated with different wavelengths of light. One group was treated with blue, another amber, and another with red. And the findings were measured: red for leaf biomass, gene and transcription of genes. Okay. Another group of samples had a control light shown on them. So what would be the most positive one? It would be sunlight because it comprises a mixture or different proportions of each of these lights. Accuracy of 2 changes. In blue, photosynthesis is greater, and in red, photosynthesis is greater as well. In blue, biomass is greater, and in red, biomass is greater as well. Right, understood. Explain the results shown in the graph. So the rate of photosynthesis is greater in red and blue light because the chlorophyll of the plants can absorb this red light, and hence as a result rate of photosynthesis is high as well. And if the rate of photosynthesis is high as well, then glucose production is high as well, and this would cause the biomass to increase rapidly as well. Biomass to increase rapidly as well. And in control plant, the growth would not be as greater because photosynthesis rate would be slow, and hence glucose would be produced less rapidly, and hence biomass would be low as well. So I have written this down here. How many questions left? Oh, two more. Okay, two more to go. The table shows that the table shows pigments contents of the leaves. This is in control light. This is in blue light. Each of the pigments, amber, red. In red light, it is the greater. It is the greatest. The red light, it is the greatest. Okay. So this question is about describe three conclusions that could be made from the results. So three conclusions that could be made from the results is that in each of the light, in each of the different wavelengths of light, there's this present of each of there's this present of each of these pigments, because in different wavelengths of light, there's this present of each of these pigments, and apart from that, chlorophyll A is highest in amount in each of this, and anthocyanins is lowest in amount in each of this. So these are the two conclusions that I could make from this table. I have written this down. And the last part is that plants treated with blue light had an increase in photosynthesis rate. Blue light has an increase in photosynthesis rate. Light dependent stage. You have to link this with the light dependent stage of photosynthesis. Make sure to remember that. You have to link this with light dependent stage of photosynthesis. Photosynthesis caused by increase in transcription of some of the genes. So just two genes whose transcription could have increased the presence of, whose transcription could be increased in the presence of blue light. So two of the genes would be chlorophyll A. First thing is that chlorophyll A. Chlorophyll A absorbs blue and red light. Chlorophyll A absorbs blue and red light. So the genes responsible for the production of chlorophyll A would be higher as well. And apart from that, the gene, I believe I did the gene coding for electron transport chain, as I showed in the earlier part, would be genes for the production. So transcription for each of these genes would be so genes for the production of electron transport chain. So yeah, you could also say something about Rubisco, that is the light independent stage, because products that are produced in light dependent stage impacts the light independent stage as well, because in light dependent stage you have the production of ATP, and in light independent reaction you utilize this ATP to make up glucose. So safest choice would be genes for the production of chlorophyll A and genes responsible for the production of electron transport chains as well. If you did like this video and if you find this video to be helpful, then make sure to like and subscribe. And if you do have any other questions, or if you're facing problems in any of the concepts, or you do need notes for anything, make sure to comment below. Make sure to comment below, or reach us through our Facebook page. The link would be attached. And yeah, if you do want to get connected with me, then you can knock to the official page of ours. Thank you. Have a great day. All the best for your exam.